Hi, everybody. Welcome to our lecture today, where we are going to focus on Mendelian genetics and how one gene is passed on to their offspring. So here is some of the learning outcomes. Some of the keywords that we're going to focus on today is, again, the concept of Mendel, looking at the word heterozygous versus homozygous, word dominant versus recessive, understanding the difference between phenotype and genotype, and then diving into the laws of Mendelian laws. There are three of them, what those mean, and then eventually work our way through the Punnett square, where we understand how we can predict what the offspring will look like based on the parental genotype. So when we're talking about Mendelian genetics, again, Mendelian genetics is a one gene concept. So most of the genes that we have in our body are considered to be genes that are not going to create um, a specific phenotype or characteristics on us. Rather, there are multiple genes that work together to present a phenotype. Now, in terms of the inheritance and Mendelian inheritance, we are focusing on a single gene. Now, looking at a little bit more detail, we do have these a specific database, which is called OMIM, or Online Mendelian Inheritance in Men, where there is over 8,000 single gene disease and traits that are being identified. And these 8,000 single genes disease corresponds to about 23,000 genes. And then we have the fourth World Health Organization, or WHO, that estimates about 10,000 diseases occurring caused by single genes, which are called monogenic. Mono again means one, as in one gene. And roughly about 1 in every 10 people in the US has some sort of rare disease, as do looking at globally about 350 million people. So this basically means, again, looking back at what the numbers imply is that we do have single genes that actually take a specific action in our body. And if those genes have some sort of mutations in them, in many cases, they can cause major health disease. As it mentioned earlier, if we have 8,000 single gene disease, they come from corresponding to 23,000 genes in our body. Now, characteristic of a single gene disease, again, so we're talking about one gene in your entire genome gets modified or mutated, and that mutation causes the disease. So this single gene disease, there is in families, the probability can be deduced knowing how it's affected the person. And again, we'll kind of expand on this as we look at the Punnett square. We can basically figure out what the offspring chances of having that disease looks like by looking at the parental genome and basically looking at how that affects the population overall. And in some cases, this disease, and we're going to put that in quote, fixable by compensating for another abnormal instructions. So basically something else might be able to fix the problem, some other gene. Now, because our focus for today is going to be on Mendelian inheritance, we're going to look at some of the vocabulary that's going to be relatively common as we discuss this concept. Now, one of the things that we have understood so far is that when you're looking at the genetic information of a person, we have two copies of the chromosomes. One comes from your mom and the other one comes from your dad. And basically every gene on that genome of that person has two copies. Now, the variations on those genes are identified as alleles. So as at this point, anytime I use the word alleles, we're basically talking about variations within the same gene. Now, we're also going to use the word dominant versus recessive a lot. Now, dominant, as the word implies, it's basically the version of the allele that's going to kind of overrides or overshadows the effect of the second allele. So the second allele would be recessive because unless you have two copies of that gene, it's not going to show any characteristics, versus dominant version, you will see the effects of it even if you have a single copy, since we're using the word dominant. Now, you also can be a carrier of a certain conditions or certain disease. And that's basically when you yourself do not show any symptoms of that condition. However, you can pass that quote unquote disease allele to your offspring and cause that child to have the offspring or may not. Now, another thing that you have to be careful, specifically when answering questions about Punnett square or Mendelian genetics, is the concept of genotype versus phenotype. So genotype is basically an organism's allele, and I'll talk about that in just a minute. So the variation in that specific gene that you're discussing. Versus phenotype, as I mentioned already, is an outward expression of the allele combination. So basically, if you have a specific characteristics, as I would mention, things like hitchhiker's thumbs, a widow's peak, your things like your eye color, hair color, skin color, if you can eat milk, if you are if you have a tendency to develop Huntington disease or cystic fibrosis. So all of these are going to be the phenotype, which is a literally outward expressions of what those alleles will do to you. Now, if you observe a trait, so again, things like widow's peak, darker skin tone, having blue eyes, having brown eyes versus blue eyes, examples on the pea plant that Mendel did, having green versus yellow. So the trait that is going to be represented will be identified as your dominant trait. For this one, anytime we want to represent a dominant allele, we represent it with a capital letter. For instance, in this case, we use the letter T. And if you have a recessive gene, which is the one that is masked or basically overshadowed by your dominant gene or allele, we will represent that with a lowercase, for instance, in this case, T. Now, because we are a diploid organism, which means we receive a copy from our mom and a copy from our dad, anytime we're talking about a gene, we will have two copies of that allele. Now, if those two copies that you're receiving from your mom and your dad are identical to each other, we're gonna use the word homozygous. And again, homo means same. So homozygous means same identical alleles. Now, that option in terms of homozygous could be both parents are giving you a dominant version of that allele, which makes you a capital T, or both parents are giving you the lowercase or the recessive allele of that gene, which we represent with lowercase t and lowercase t. Now, heterozygous implies that each parent is contributing a different versions of the allele. And that in this case would be a capital T and a lowercase t. So one of the two parents, doesn't matter which one, is contributing a dominant copy of the allele, and one parent is contributing a recessive version of that gene. Now, as we talk about the Mendel's experiment, I'm going to bring up the three main laws that basically Mendel has discovered based on his understanding of his research and his experiments. Now, one of the things that is really cool is when Mendel was doing his experiments, it was happening a long time ago. So we're talking about mid 19th century, so 1857 to 1863. And there was no concept of DNA. If you guys recall, the concept of DNA, double helix, inheritance, it started to come into play later on in the 20th century. So he didn't really understand what the DNA or cell or chromosomes even were, right? But he used his pea plants to explain how a specific traits are being passed down from the parents to their offspring. So it's a transmission of those kind of characteristics from the parental to the offspring. And he only observed it in diploid species, which implies that you must have a maternal and a paternal side of the genetic. Well, I should say, you should have two copies of the genetic information coming from the both parents, maternal and paternal. Now, Mendel also deduced that the consistent ratio of traits in an offspring indicated that plants transmitted these distinct units. And you're like, what does that mean? So as we look at the Punnett square, it becomes more clear that when we're talking about the specific genotype alleles that get passed down, we have a very specific units of transmission, and we can actually literally calculate and figure out what the genotype and what the phenotype of that person or that specific characteristic will be. Now, Mendel experiments, for us to understand a little bit better, I'm going to kind of introduce a concept called true breeding. True breeding is when an organism with the same alleles or genomes produce the offspring that has the same trait. Simply put, that means that the offspring has the same characteristic as the parent. For instance, a short plant produces short offspring. Now, as we study, and I'm not really going into details of how Mendel presented or carried out his experiment, rather what he understood from his experiment and what organism he used and what part of that organism he actually focused on. So when we're talking about Mendel's experiment, one of the most important things that he deduced from his experiment is what we call three main Mendelian laws. They're called law of dominance. So we already talked about the concept of dominance, right? Overshadowing a recessive gene, law of segregation, and law of independent assortment. So let's take a look and see what each of these laws actually focus on. So law of dominance, again, we're kind of going back to that concept of one version of the allele will overshadow the effect of another allele. And again, this is very simple inheritance where only one gene is involved in creation of a specific trait. So the law of dominance states that in a simple inheritance, again, one gene being affected, the dominant allele is always going to override or overshadow the other allele, which we always identify as the recessive version. Now, I have already talked about some of the examples with you, but here is kind of a depiction of what that is. So Huntington disease is considered a dominant trait, which means that if you have only one copy of that genetic information, that would be enough for you to develop that disease. So Huntington disease is something that a lot of people are afraid of. It's a very degenerative disease that affects the nervous system. Unfortunately, if one of the parents is diagnosed with Huntington disease, the chances are that the child also will have that disease. We also have what we call widow's peak, which is demonstrated right here. So the presence of widow's peak is depending on only one copy of that gene being present to create a pattern. Versus, and also another example would be the hitchhiker's thumb. So the normal thumb is the dominant pattern. So where your thumb is more straight if you're holding it up, and then the hitchhiker thumb would be the recessive gene that's basically overshadows. So it's the straight thumb versus the hitchhiker thumb. Now, we know and we understand so far that if you're looking at a diploid organism where the parents are providing the genetic information, one copy of the gene, and if one of these genes is identified as the dominant copy, which is the capital letter, you only need one copy being passed down from the parents for the child to exhibit the condition, the symptoms, the characteristics. Now, as Mendel developed his experiment, he noticed something else. And based on the understanding is he came up with a law of segregation. Now, what does law of segregation implies? It implies that, as the word segregation implies, means separating. They say that the parent only give one allele of their genome to each of their gametes. And remember, the word gametes means the sperm and the egg. And this is happening during the meiosis process. Now, again, we had a full lecture on meiosis, so that should not be something new for you guys, but I wanted to still demonstrate what that means in terms of meiosis by looking at this picture. So in this case, the first parent, let's say this is the maternal based on the color pink, it's a heterozygous organism or heterozygous characteristic, where one is the dominant gene and the other one is the recessive gene. For the second parent, you have a homozygous dominant where both is represented with the capital T. Now, as we go through the meiosis, so interphase 1, you double this genetic information, right? So you have now 2 capital T and 2 lowercase t, and then for the second parent or the paternal parents, you have all capital. Now, only one copy gets passed on by the end of meiosis to each of them. And this is completely random, basically. Each genes only gets passed down one of these copies. So by random, it goes one copy of T to this one cell, the other copy to this cell, and this cell, and this cell. And now these parents, because all of them were capitalized or homozygous dominant, every cell would receive a copy of the dominant. Now, as we look at the gametes being combined, there were 2 possibilities of the gametes being produced. For the first possibility, each of the parents are going to pass a dominant allele to their offspring. So let's say it's passing this one and this one. If both of them passing out a dominant version of that allele, you get the first example during fertilization, and that offspring will have a homozygous dominant characteristics or genotype, I should say. However, if this parent passes the lowercase t and the second parent passes the uppercase t, then you end up with a heterozygous genetic setup where you have, again, 2 versions of the gene, with one being a dominant, the other one being a recessive. Now, again, this experiment basically confirms that the hybrids hide one expression of the trait, which reappears when the hybrids are self-crossed. So we look at this concept in just a minute as we look at the Punnett square and how those characteristics gets passed on. Now, self-crossing is a new word if you're not familiar with it. Literally, it means that you are using the male genetic information of the plant to refertilize it. So basically, the genetic information from both parents come from one plant. It doesn't come from two separate plants. So let's take a look at an example of these Mendel's experiments and get familiar with some of these concepts related to the law of segregation. So in this case, anytime we use, moving forward, we use P1, that implies parental generation. F1 stands for first filial generation, or basically first line of offspring. And F2, it stands for second filial generation, which is the second set of offspring created from the F1. Now, wild type, which is going to be the most common phenotype, can be both recessive and dominant. And wild type is basically what we're going to see, again, most commonly within your environment, or it would be the trait that if you look at a general population, it would be the most common phenotype in that population. We also already talked about monohybrid crosses. Monohybrid crosses, you only concern about one specific trait. So in this case, if you look at the example on the paper, you only see we're focusing on the height of the plant, a tall plant versus a short plant. And as we look at this example, I want you guys to understand that the tall plant or having tall phenotype is the dominant allele compared to the short plant being the recessive allele. So in this case, if I am to write the parental, remember the true breeding. This would be, because I'm doing true, this would be capital T, and the short plant will have a lowercase c, lowercase t. So again, this parents would be a homozygous dominant, and this parents would be, homozygous recessive, right? Because both of the alleles have the same copy. Now, when I cross this, all of the F1 generation will be identified as tall plants, because again, the capital T, it will be passed down to all the kids from the first parent, and therefore from the tall parents, I should say, from the tall parents, and all of the kids will develop that dominant trait. Now, as F1 starts to grow, what we can do is we're going to do self-crossing. So self-crossing, basically, again, as I implied, it basically you are fertilizing yourself. So both genetic set of information comes from one parent. And when you do that, when you create your F2 generation, one thing that you notice is that you have out of the 4 possibilities, you have 3 of them being tall, and 1 out of the 4, or 1/4 of that, would be the short. So, and I will look at why there's 4 here. This is called associated with the Punnett square. But here's what I want you guys to understand. If I'm looking at the words that are here, and we'll explain where these come from. When we talk about the short, that means that it's fully homozygous recessive. When I said 1/4 is tall true breeding, it means, again, it will have the same trait as the parents, so capital T, capital T. And then half of this, or 1/2 of it, is non-true breeding, which means they're going to be heterozygous, where they have the dominant trait, but they also have a second copy of the recessive gene present in their genotype. Now, for us to kind of look at the Mendel inheritance and law of segregation, we need to understand where these what are the traits that actually Mendel took a look at. And here is the main traits that have been observed by Mendel, where he draw his laws from. So he looked at seed form, seed color, pod form, pod color, flower positioning, color of the coat of the seed, and then the stem length. So stem length is what I just talked about, the short being the recessive one and tall being the dominant one. But look at this, guys. You can see it. The picture does a really good job for you to distinguish these single gene traits or monogenetic inheritance. So round is going to be your dominant trait over the wrinkle. The yellow pea is going to be the dominant gene over the green. Inflated, kind of chubby pod form versus restricted. We also have the green pod being dominant over the yellow pod. The flowers being along the stem being more dominant characteristics than the flowers being at the end of the stems. Having a gray or gray-brown seed color over white. And then lastly, plants that are tall are dominant over short plants. So again, these are 7 characteristics that are controlled by single genes. And if you have a dominant trait that are shown on the first row, those are the ones that are going to be more obvious in the general population. Now here's a table which uses the law of segregation to figure out the ratio for the dominant versus recessive. Is it something that you guys need to understand? And no, a lot of my students wanted to see, like, when I originally discussed this in person, they wanted to kind of see it in numbers. So this is what they look like. So I just presented you the numbers. It's not something you will be tested on. So you can kind of skip over these. Now, we have been talking about the law of segregation and how a parent passing one of the copies of their genetic information to their offspring. And basically, when you get and create your offspring, each parent has contributed one copy of the genetic information or allele. Now, Punnett square is going to give us an opportunity to understand, based on the genotype and the phenotype of the parent, what are the genotype and the phenotype of each child going to be. So it helps us really calculate the frequency of the genotype and phenotype for the filial 1 and filial 2, and so on and so forth. So you can basically trace a specific characteristics being passed down if they are single trait genes. So in this example, the paternal genotype or father's genotype is depicted on the top. So we place them on the top of our little square. And then the maternal genotype will be placed on the y-axis of our little Punnett square. And in this case, to just kind of, again, reiterate some of the vocabulary, the parent is a heterozygous dominant. So hetero as in 2 versions of the gene, and then the mother is a homozygous dominant. And all you need to do at this point to complete your Punnett square is kind of multiplying these specific alleles from the paternal with the maternal. So what does that mean? So let me just change my color pen so I can show you. So if I put this one and this one together, I get R and R, or homozygous dominant. If I combine this gene with this, kind of doing like a multiplication in another sense, R would give us this. If I come down all the way, and this one, again, gives us homozygous dominant, and here to here gives me a heterozygous dominant. So if I was to look at this Punnett square and say, okay, these 2 parents have the genetic information of heterozygous dominant and homozygous dominant. What is the genotype of the offspring? And what is the phenotype of the offspring? Okay, so let's say R corresponds to the seed being round, if it's capital R being the dominant, and if they are wrinkled, it would be the lowercase r. So that's for the phenotype. What is the genotype of F1 based on this information? So you have 1 and 2 out of the 4 squares. So 2 out of 4, which is the same thing as saying 1 out of 2, is identified as RR. And then the other half is heterozygous dominant. So again, 2 out of 4, which is the same thing as 1 out of 2, are heterozygous dominant. So that's identified as your genotype, right? The genetic variation of the alleles for the F1. Now, because the R is the dominant and I only need one copy of this, all 100% of the cells would be a round phenotype because it's either homozygous dominant or heterozygous dominant. So both of these genotypes produce the round phenotype. Here is another example of the color brown eye. I do want to point out that in reality, the brown eye, blue eye concept is not a single gene, but I use this because it helps you a little bit better visualize the concept. But in reality, if we look back at ourselves, our skin color, our eye color, our height, they're not controlled by a single gene. So it's not as simple as it's depicted here. So please do keep that in mind. And I believe I do have a slide on that as well. So right here, we're gonna say that the brown eye is the dominant and the blue eye is the recessive. So again, when we're talking about dominant characteristics, either brown eye color is either created by the heterozygous or the homozygous dominant pattern of genotype. And blue eye will be produced by a homozygous recessive genotype. So here, this person has blue eyes, which means again, it's capital B, sorry, lowercase b, and the maternal has brown eyes, which they depicted as capital B, because they are true breds. So we're saying basically you are breeding a homozygous dominant with what? A homozygous recessive. So homozygous dominant and homozygous recessive. So again, each of the parents contribute one copy of their genetic information, right? So the maternal side on this side will be contributing a dominant allele, and the paternal side will be contributing a recessive allele, which means, again, 4 out of the 4 chances are that the offspring will have a genotype of capital B, Again, it means 100%. 4 out of 4 means 100%. And the phenotype, because it's a all heterozygous dominant, would also be 100% brown. So that's another way of writing it. 4 out of 4 versus 100%. And for those who might be a little bit, I guess, struggling with the statistics and converting percentage to actual data, this is what we're looking at when we say 1/4 is the same thing as 25%. If we say 1/2 or 2 out of 4, that means 50%. 3 out of 4 means 75%. And then 4 out of 4 implies 100%. So I can depict this data in both ways, either as a fraction or as a percentage. So do keep that in mind. Now, this is an example we already take a look at. We have used different letters. So I'm not going to repeat the same information, but we talked about the height of the pea plant done by Mendelian inheritance to be a single gene phenotype, which means that you only need one gene affecting this trait, and you only need one copy of the allele to be dominant to represent those characteristics. So in this case, you're using a true bred, which means a homozygous dominant with a homozygous recessive. You cross them to create your filial 1 or F1 generation with a genotype of 100% capital T, lowercase t, or again, 4 out of 4. And then phenotype, all of the plants will be tall because this is heterozygous dominant. Now for the F2 generation, sorry about that. For F2 generation, we're going to use the self-crossing, which means, again, the genetic information is coming from one parent. So because 100% of the genotype was capital T, lowercase t, for both paternal and maternal, we will have the same identical genotype. And again, just do that kind of a crossing. So you do this, you do this, and then finally this. So what you end up with, sorry about that. So what you end up with is 1 out of 4 is going to be capital T, 1 and 2. So that's another 1/4, that's another 1/4, and that's another 1/4, right? So 1/4 or 25% would be capital T. 1 and 2, both of these are heterozygous dominant, so 2 out of 4, or 1 out of 2, or 50% is going to be heterozygous dominant. And then the last box, which is homozygous recessive, will consider 25% of the F2 generation. Now, phenotype, again, look at this. This is a dominant trait being tall. So out of these 4 boxes, this one, and this one will show a characteristics of being tall plants. So 3 out of our 4 plants, or 75%, have the phenotype of tall. And the only one that's going to be the short plant would be the homozygous recessive copy, which is 25% or 1/4 of it. So when you guys, again, I want you guys to point out here again, same thing we discussed. 1/4 is going to be short, and 3/4 are going to be tall. This one says true breeding, implying they're both capital, and these two here would be tall non-true breeding, which means they are heterozygous. And the first one are short, which means they are homozygous recessive. Questions that you will get in an exam for this, guys, focuses on you looking at the F2 or F1 generation and being able to distinguish what is the percentage for each genotype. And based on that genotype and the fact that a gene is dominant, you have to figure out your phenotype. Now, here's the second thing that we want to kind of take a look at. In this case, you have a tall plant and you have a short plant. Now, we're not going to talk about the short plant right now. We're just going to focus on the tall plant. Let's say I give you a tall plant and I say, tell me what is the genotype of this plant is. Now, because the tall plant must have the phenotype of tall is associated with a dominant gene, that means you only need one copy of the allele to create a tall plant. So the genotype of this one right? The genotype of a tall plant can be a homozygous dominant or a heterozygous dominant, because all I need is one copy of the capital T for my plant to represent a phenotype of tall. So how do I resolve this issue to figure out if my parental genotype, or this plant's genotype, is homozygous dominant or heterozygous dominant? Easiest way to accomplish that is you're going to cross test or cross them by doing a crossing with a homozygous recessive gene. Homozygous recessive gene. So here are the possibilities. If my parent that had the unknown genotype has capital T, which is a homozygous dominant genotype, all of the plants are going to be what? Tall when I crossbreed them with a homozygous recessive. Because regardless of, look at your Punnett square, regardless of what the recessive gene is contributing, I always going to have one copy of my dominant gene, which means you will end up with 100% heterozygous dominant, right? Which means all of your plants are going to be tall. But if I cross this with a recessive, homozygous recessive, and your genotype is capital T, lowercase t, then you end up with this. So half of your genetic information will have a genotype of TT, and the other half will have a fully recessive gene because the parents are both have a copy of, at least one copy of the recessive gene being passed down, which means half of those will get 2 copies of the recessive gene, and therefore would represent a true-bred plant with short characteristics, and 2 are going to be heterozygous dominant with a tall characteristics. So see, just by us doing a crossbreed with recessive, we can get 2 very different outcomes. Either fully tall phenotype, everything is tall, versus half and half, where you have, again, half representing a true bred of homozygous dominant and 2 representing a homozygous recessive characteristics. So this example and doing a test cross, we always going to use a homozygous recessive or true bred recessive plant to figure out the possible genotype of the first plant. Now, again, we talked about the single gene inheritance, right? We have spent the Punnett square. We talked about the fact that Mendel had looked at 7 characteristics that are considered to be dominant compared to their the other allele being recessive. But in reality, if we're looking at humans, the single gene inheritance is not that common. And the disorders from that single gene is also very uncommon. Now, let's take a look at this. So, for instance, phenotypes associated with the single gene are always influenced by other genes and also by some environmental factors. So we're talking about epigenetics. For instance, I talked about the eye color earlier, right? The brown color being the dominant gene as either homozygous dominant or heterozygous dominant. And blue eyes being, again, I'm just going to write it so you guys can hopefully remember. So you can have this, or if you have blue eyes, then you have homozygous recessive. But even though this is a single gene representation, we have evolved to understand that our genome is more complicated, and therefore other genes can also influence our eye color. So How does our eye color actually differ? One of the concepts that actually affects our eye color is how much melanin and how much of melanosomes do we actually create. So melanin is the one that basically gives you your skin color as well. So it's a dark color pigment that's present in your eye and skin, and the more of it you have, the kind of a darker skin color or darker eye color you will have. Melanosomes are the structures that carry melanin. So again, the more of these melanin-packed melanosomes do we have, the darker the color is going to be. And the surface on the back of the eye, the iris also contribute to the intensity of the color. So melanin is also present inside the eye, inside of your eye. So when you have the light hitting the inside of your eye and reflecting back, it's also contributing to how intense they are. So the more melanin you have internally, it also reflected out and gives you kind of a more intense eye color. Now, one of the interesting things that I wanted to talk about here, and I'll show you guys on the picture in a second, is there is a main gene that controls the amount of melanin being produced, and that gene is OCA2. So OCA2 is a dominant gene. When you have one copy of that gene, your eyes are going to be brown. However, a second gene is also involved. It's called HERC2, and it controls the expression of OCA2 gene, which is the one that gives you the brown eyes. So what does that mean? Let's look at this example. So OCA2 is a gene that basically implies you have brown eyes. It's a dominant gene. So if you have one copy of it, you will have brown eyes. So look at the first one. In somebody who's albino or has albinism, you have no gene. So there is basically either a defect or a missing gene. So you have no copy of the gene being passed down from the parents to the child. So they have no color inside their body, no melanin, no skin tone. Another option is when the OCA2 are both recessive. So both parents are contributing a recessive copy of that allele to their child. So again, we represent it with lowercase b. If you have brown eye, you can have dominant and recessive being passed down, which means one parent contribute a dominant version of the allele and one parent is the recessive, or both parents contribute a dominant version of the allele. And I'm sorry. So that gives you your brown eyes. However, if you have the expression of the HERC2, then you actually present the blue eye because it marks both of those, the OCA2 gene as basically no effect. It abolishes the activity of that gene. So it's literally kind of taking away the brown eye color that is being given as either a heterozygous dominant or homozygous dominant from the parental to the child. Now, these genes are both near each other, which is located in chromosome 15. So is there a chance basically that you have both parents having brown eyes and you have a child that is blue eye? Yes. Is it as common? No, but it is a possibility. Now, we have talked about the concept of sex chromosome versus the autosomal chromosomes in detail in our previous lectures. We know that the sex chromosomes are identified as X and Y. Women or female have a genotype XX, which means both parents contribute a copy of the X to create a female offspring. And the male is represented with the letter XY, which means that the offspring has received a Y copy of the chromosome from their paternal side. The father is the only one that can give you the Y. So when we're looking at sex chromosomes, you have a specific inheritance pattern that comes from the X and Y chromosome. Now, we have a full lecture right after this one about the X-linked genes. And we talk about X-linked mostly because Y is a relatively short chromosome and doesn't really contribute much, versus the X chromosome is extremely important in terms of the health of the individual. And there is a lot of medical conditions that is associated with the X chromosome mutations. So we'll talk about that in the next lecture. What I wanted to mention here is the autosomal chromosomes. Autosomal chromosomes are basically the remaining chromosomes that are not identified as sex chromosomes. So if we have in a human, we have 23 sets of chromosomes or pairs of chromosomes. 22 of those will be identified as the autosomal chromosome, and one pair would be the sex chromosome. Now, there are, again, some very specific medical conditions that are associated with these autosomal chromosomes. These medical conditions can either be what we call a dominant traits or recessive traits. So autosomal dominance is something that we talked about. Again, you only need one copy of it for that disorder to be represented. One of the worst kind of dominant copies you can receive in terms of disorders is a copy of the Huntington disease gene. So Huntington disease is considered a mutation, and you need only one copy of that mutated gene to be affecting the person. It is equally affecting both male and females because, again, it's not an X-linked medical condition. Autosomal recessive implies that you need two copies of the recessive allele to be present for you to develop that medical condition. And in that case, autosomal recessive, cystic fibrosis comes to mind, where we talked about, I think, cystic fibrosis before in this class. This is a really tough disease. Kids usually don't get to be that old because there's problem mostly with the respiratory system and other organ systems. But there's really built up of significant amount of phlegm and pus within the respiratory system. But autosomal recessive, usually the way it's observed is that it skips one generation because those are going to be your carriers. It means they carry a copy of the recessive allele, but they don't represent any of the symptoms. This is a really good way of you guys comparing this autosomal dominant versus recessive. So again, autosomal dominance affect male and female at the equal level because it has nothing to do with the sex links that may contribute to it being highly presented in one sex or one gender. In this case, they both get affected equally. Autosomal recessive also have the same frequency because, again, it's not an X-linked disease. Autosomal dominant, because of its dominant characteristic, does not skip a generation, which means every parent will pass that characteristics to their child, versus the autosomal recessive can skip a generation, not will skip it, can skip a generation. So you may see it in the grandparents and then the grandchildren. Affected individual has an affected parents. Again, so if your parent have it, you will have it in your children, and unless there is a de novo or a new mutation which prevents that from being passed down. And then affected individuals has parents who are affected or are carriers. So you could have a child that will have the disease And one of the parents may have the disease, or both of the parents may have no symptoms of the disease and only be the carriers of it. So if that's not clear, so basically what I'm saying here, you could have a child that is basically, let's say, TT, okay? That says for cystic fibrosis, okay? Well, maybe I write it as lowercase f for cystic fibrosis child. So the parents can either be FF crossing with FF, so the little lowercase f was passed, or you could have a parent like this. So a parent that is fully affected and a parent that is a carrier. So this is an affected parent, and this is the carrier. And they will produce an offspring that is either a affected person or an offspring that is a carrier. So this is P1, this is F1. Sorry about that. Now here's a cool thing, guys. We can solve genetic information looking at this concept of Punnett square, looking at the population, tracking the genetic pattern from the parent to the children to the following generations, and basically see what is the chance of a child developing those disease or symptoms based on those Punnett squares. So one of the, on the meaning of the dominance versus recessive, it is important to understand if the dominance is something that causes the disease, or is it the recessive version that is causing the disease? Kind of like what we were talking about, the cystic fibrosis. For instance, cystic fibrosis, you can have a carrier, but no symptoms of it, versus if it's a dominant gene, you will have that time passed on from the parents to the next generation. Now, recessive traits are due as a result of loss of function, which means the disorders tend to be more severe if you have a recessive trait, so something like cystic fibrosis, versus dominant traits are usually as a result of gain of function, which means you, and I'll show you this on a picture. So in loss of function mutations, and that basically cause the change in the genome, if you have a DNA being affected, so here in this case, the DNA is being affected, a mutant is formed, we create no protein that leads to enzyme deficiency. This is a representation of a recessive concept. So loss of function, you completely lose that protein, versus gain of function for a mutant. So in the case of your Huntington disease, you are gaining a function by having, or as I said, gaining the disease by having only one copy of that medical condition. So if you have a mutation in that genome, you gain the function back because there's damage to that dominant gene. So less chance of you being passing it down, basically. Okay, law of independent assortment. So we mentioned that Mendel's have three major laws. We talked about the law of dominance, we talked about the law of segregation, and the last one is the law of independent assortment. I did hint at it before, but just to kind of looking at it in terms of meiosis, independent assortment means basically a specific chromosome that gets passed down to the gamete and then eventually used for the fertilization of the egg happens completely randomly, and it doesn't really follow a specific pattern. So here in this picture, they're showing the process of meiosis 1 and meiosis 2. And notice that there is two different alternatives. So alternative 1, notice that they're the same thing. So here, in this case, everything is the same, right? But the only thing that's different is the way that these chromosomes are organized. So the way they get passed down at the end of meiosis 2, this one, let's say you got copies from the paternal, no maternal genetic information is passed down, and these two cells got completely the maternal information. Versus this cell, each cell, or each gamete, I should say, has both maternal and paternal information. So you can see this one based on how the genetic information was sorted as it gets passed on during meiosis. You either got the chromosomes from the paternal side and nothing from the maternal side, or a mixture of those. So to just kind of help you understand this a little bit better, sometimes I tell a student, think about certain characteristics that you get more from one parent versus another parent. And that's basically, you may have had a higher contribution of those gene during meiosis coming here, and that parent will have the more dominant characteristics. But again, independent assortment, all it means is when we undergoing the process of meiosis and where the genetic information is being randomly pulled on opposite sides of the cell, you literally doing this job randomly. There is nothing that say all of the chromosomes from the mom go to this side, all of the chromosomes from dad come to this side. You kind of mix them, you line them up in the middle and take half of them to one side and the other half to the other side. Now, the examples we have worked so far on, they're all considered monohybrid crosses, which basically meant we only looked at one specific characteristic or trait and track it to see how it passed on through F1 and F2 generations. Dihybrid crosses is going to look at two separate traits and how those separate traits are being passed forward. For instance, if you think about the 7 characteristics of that Mendel has studied, one of the study was, is the piece going to be round or wrinkled? And round was the dominant version. So this is again, round was dominant, so we will represent it with capital, and then if it was wrinkled, represented with lowercase r. Another characteristic that was observed was the color of the pea. So yellow was the dominant, and if it's not, then green would be the recessive. So at this case, we're actually looking at the pattern of inheritance for both of these genotypes at the same time. So we're using a concept of dihybrid. Again, 2 different genes and how they get passed on. So you start with a true bred. So you have this one having the dominant characteristic, which means it's going to be round, capital R, and the color is yellow, which means capital Y. And then the other side, you will have a homozygous recessive for both traits, which means it is wrinkled, lowercase r, and then green, which is, yes, that's the recessive version of the gene, and that would be lowercase y for both allele. So they all produce round yellow, which means if we were to write it, it's a heterozygous dominant F1 generation. And if we're looking at the F1 crossing, so right, self-crossing the plant, it means you're doing a heterozygous with a heterozygous dominant characteristics. So same pattern, nothing is different in this case. You now have just the 2 separate genes that needs to be written. So R's go together, Y's goes together. So this is capital R, lowercase y. This one is uppercase R, and then lowercase y, which gives you this one. So literally do that kind of a concept of multiplication, put the letters that are the same together, and then figure out based on that genotype. Now, there is a lot of different genotypes here. So you can look at the genotypes to basically understand that there is genotype where you have R, R, Y, R, sorry, I'm just going to say it, Y, Y. You can have genotypes like R, R, Y, Y. You can have R, R, Y, Y. And I'm just giving you some of you to see the variation of it. I'm not going to go over all of them. So when you look at the phenotype, which is something that I want you guys to know, the phenotype, because these genes are dominant characteristics, I don't really care what the second positions are, okay? Because the second position can be capital or dominant, or it can be recessive. But regardless of if the second allele is dominant or recessive, I will still exhibit the phenotype that is the dominant phenotype. So for instance, what does that mean? So in the first example right here, I can have R, R, or R, R, and then Y, 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 Y. And all of them will give me a round yellow P. So it doesn't matter which of these two copies I get, they get the same thing. So that little dash line, it means the second copy can be either of the alleles, recessive or dominant. The final phenotype will be the same. So this one is a round, which means I get dominant trait, but the color is the recessive. You can have a recessive for both, recessive for the shape of the cell, so shape of the P, I should say, and then the dominant for the color, and then all recessive, homozygous recessive all across. So the trait's basically going to be a ratio of 9 to 3 to 3 to 1 for the phenotype, which means if you say, okay, what is that 9 to 3 to 3 is? So 9, 3, 3, 1. This means homozygous, and I'm not going to spell the whole thing, homozygous or heterozygous dominant for both. Okay? This one is homo for, I should say, homozygous dominant and homozygous recessive for 3 of them. Same thing, homozygous dominant, homozygous recessive. I should say homo or hetero, sorry. Hetero, and then the last one is homo recessive for all of them. And again, hopefully this is not too confusing. Look at the characteristic again. Both of these can be homo or heterozygous dominant, which is the first one I wrote here. The second set, which means 3 out of the 16 boxes that I have, could be homozygous for one characteristic being recessive, right? Homozygous recessive, right? And for the second characteristic, it could be either homozygous dominant or heterozygous dominant, which is these 2 examples that I have here, right here and right here. And the last examples, which is the rarest of them, is having all recessive alleles, which means they both represent the phenotype recessive for both characteristics. So 9 out of 16, 3 out of 16, 3 out of 16, and then 1 out of 16 will have the same characteristics. So the way we represent it is 9, 3, 3, 1. I'm gonna write this one more time, so make sure everybody follows this, because I don't want to confuse. Sorry, if I did confuse anybody, that was not the goal. So the 9, which is the highest, is homo or hetero dominant for one characteristic, and then homo or hetero dominant for the second characteristic. Okay. 3 is homo or hetero dominant for one characteristic. Okay. And homo recessive for the second characteristic. The other 3, same concept. Homo recessive for one characteristic, and either homo or hetero dominant for the second characteristics. And then 1 would be homo recessive for all, homo recessive for all characteristics. So if I add that, it would become 16, which is the size of the Punnett square you see right here. Hopefully that makes it a little bit easier. And then this is corresponding to this one, this thing, this one, and then lastly this one. So this is a 9 to 3 to 3 to 1 ratio. Okay. So probability. So this is kind of thinking about the statistic, the likelihood that an event will occur, right? Kind of simple. So the probability of a simultaneous independent event equal to the products of their individual probability. Like, what does that mean? So we'll talk about it in an example because I think it makes it a little bit easier. So we are looking at a Punnett square looking at these 2 characteristics, which is again R is represented for the round, and then versus wrinkled. I'm going to keep saying this so you guys remember it. Y is the color yellow versus green. So that's the dominant, that's the recessive, that's the dominant, that's the recessive. So basically, an example that I showed you here said, consider the probability of obtaining a plant with wrinkled green peas. So wrinkled green peas has a genotype, lowercase r, and then homozygous recessive for that, and then a dihybrid crossing between a heterozygous dominant. So this is your heterozygous dominant. And then says, I consider probability of obtaining a plant with wrinkled green peas, lowercase r, lowercase I, which is again, this would be your homozygous recessive. So if from a dihybrid parents, so we can separate each of these from each other. So notice what it says. Its probability of the simultaneous independent event equal to the product of the individual probability. So I'm going to calculate what is the chance of the parent, heterozygous dominant parents, having an RR offspring, and what's the chance of them from a heterozygous dominant parent giving rise to a homozygous recessive gene. Each of them have their own probability, right? Whatever probability I get for each of these, I multiply them to get my final answer. So again, if it's not clear, let's take a look at it. So I'm going to do a Punnett square for the shape of them, and then a Punnett square for the color. So when I do my Punnett square from the parents of heterozygous dominant, I figure out that 1 out of 4 chances are that I will have a wrinkled seed, because RR genotype is going to give me a wrinkle 6. That's the only one that's going to give me a wrinkle 6. So I write 1, 4. Okay. And then out of that, looking at my Punnett square for the color, the only one that's going to give me a green offspring or green P would be 1 out of 4 too. So it would be 1 out of 4. So I multiply these together. 1, 4, 1, 4 times 1, 4 is equal to 116. So I have, if I cross these two parents together, I have 1/16 of a chance to get a offspring that's going to be wrinkled and green in color. So what if I have 3 traits? Simple. Again, you do Punnett square for each of the characteristics. So the first characteristics is identifying the color yellow urine versus red urine being the recessive. The second Punnett square, so this is the first, second, and third. The second Punnett square is focusing on the colored eyelid versus the normal eyelid. So colored eyelids are the dominant one. And then the last one is the normal fingers versus the short fingers. Normal finger being the recessive and the short fingers being the dominant trait. So all I need to do is do a Punnett square for each of them. And I said, what type of gametes am I looking for? So I calculate them individually. So I wanted to have a child that has these following characteristics, okay? Red urine. Red urine means they have to have BB. So I look at my Punnett square. 1/2 of my Punnett square is going to have yellow urine. I'm looking for color eyelids. So colored eyelids is created by a heterozygous gene. So that's again 2 of those. So that's another 1/2. And then lastly, I'm looking for short fingers, and short fingers again is a heterozygous recessive. 2 of the portions of my Punnett square have that one. So I do this, and then I multiply them together. So I said 1.5 times 1.5 times 1.5 gives me 1/8. So if I want to have a child with these characteristics, there is 1 out of 8 chances that my child will have these characteristics. So the probability of having these characteristics is 1 out of 8. And you can, of course, write it in percentage as well. Now, this is also important in terms of us understanding the pedigree analysis. For researchers, we actually use the concept of pedigree, which is these beautiful charts of inheritance, to figure out what is happening in terms of characteristic being passed down. It's a very easy way of tracking conditions or phenotypes through generations. So they use this kind of a signing analysis. I do ask you guys some of the generic ones that I want you guys to know. For instance, this pedigree analysis, which is something, if you create it, it would be something like this, or something like this. Now, I'm not going to go into details about what they are. That's not the goal of this class. But I want you guys to understand that if you're looking at one of these, the circle is always represents a female, the square represents a male. If they are colored, that implies, or they're blackened, I should say, that means they're expressing the trait that you're interested in. If they are half colored, that means they are the carrier of that conditions, but they don't show any symptoms themselves. When you look at the line through the circles, depending on, or the squares, that means the person is deceased. And if it's a diamond shape, that means the gender of that person is unknown. If you have a stillborn babies, there is a line through them with the letter SB capitalized underneath them. The concept of pregnancy is the baby is not born, so we can write them as male or female with the letter P in the middle, etc. And then if you want to show generations, the line coming down is going into a new generation or from parent to the offspring. Parents are connected with a horizontal line. and adoption is a dash line because they are connected to that family, but there is no genetic history with them. You can skip this slide, so I'm not going to talk about this one. Importance of the pedigree for us, again, is very important for us because we can track medical conditions as we look at how they get inherited from parents to the children and what is the probability of that genetic information being passed on to the future generations. So you can calculate the probability that, let's say, 1 out of 16 chances that your kid will have this. And it allows us to, again, figure out the patterns of inheritance for specific genes in our body. This type of research is very important when you have groups of people basically staying within themselves and intermarrying themselves. So there is a chance of, if there is medical conditions, you want to see that trait being developed more aggressively in a population because of the, and put that in quote, interbreeding within a small population. Here is an example of a dominant trait being passed down from between 5 generations, starting with the parental generation all the way to the 4th generation, not 5th generation, 4th generation. This dominant trait is polydactyly, which basically means you have extra fingers or toes. And notice that, again, this is an autosomal dominant trait, which means you only need 1 copy of the genetic information to be passed along to the kids. So you can track it, that in this case, the female had the disease, the polydactyly, and both of the kids will have it as well because of the fact that this is a dominant and autosomal. Another medical condition that can be tracked is an autosomal recessive trait, albinism, which is, again, there is no melatonin being produced by the body. In this case, what they have shown is you have 2 sets of parents. You have basically ones that are normal, and normal by mean they don't have albinism. These are carriers, so that means they are normals, but they are showing symptoms. They don't show symptoms, but they can carry the gene. So each of them produced a carrier, and when those carriers meet, the chances are that their children might have albinism. If you recall, when I was talking about the autosomal recessive characteristic, I said there's chances where it skips 1 generation, and then you observe it in the next generation. So from grandparents to the grandchildren. Now, Punnett squares and the pedigree that we have been talking about is very important for us to figure out what is the chance of you begin passing that genetic problems or mutations or dominant gene to your next generation. There is an example that we have. Tanisha's brother has sickle cell anemia, which is basically that blood condition we talked about in detail. Now, Tanisha wants to see what is the chance of her kids developing sickle cell anemia if she has kids with her husband, basically. So here is the pedigree of the parents of Tanisha and Dawson. So they're both carriers, as you can see. So again, half color one means they are carriers, but they don't show the symptoms. And that caused Dawson to basically be a full carrier of sickle cell, which is lowercase s's, or having a homozygous recessive. So the parents would be like this, and he will be a homozygous recessive. Now, Tanisha basically is trying to figure out if she is a carrier. So is she like this, right? Or is she completely homozygous dominant, where she doesn't have the symptoms, and basically it's capital S, capital S. Now, Tanisha's husband, as I mentioned, has no family history of sickle cell anemia, so we're drawing it as this kind of the simple square with no coloring inside because there is no history. So they want to see, so they want to see what is the chance of their child having sickle cell anemia based on their history. So here is the probability that Tanisha is a carrier is 2 out of 3 because of the parents. So this is Tanisha's parents right here and right here. So if you count them, this box and this box are basically implying that she is a carrier of the gene. Oh, sorry, it should have been only half. I shouldn't have said this like this. Sorry, it's only half. She's a carrier in half of the situations. And then I just noticed the problem. And then if Tanisha and Antoinette basically create their offspring, if she is a carrier and her husband is fully dominant gene, then there is this probability of their child being a carrier, but there is no chance of the child having sickle cell anemia. So I'm just going to change this number. So this is actually 1/2 from the parents, and then half from her and sorry, I just noticed that I did something majorly wrong here. So total probability is 1/2 times 1/2 because it's from the parents to the next generation. There is no 2/3 here. So there's 2 sets of generation that are being passed down. So it's 1/8 chance of 1/4 chance of the child will having sickle cell anemia. I apologize for that. But again, all you need to look at is the probability of the parents that has passed it to Tanisha and then doing Tanisha as a carrier and writing it as the husband being the dominant homozygous and then multiplying them together. That gives you about 1/4 chance of Tanisha child being the carrier, but not to have the actual sickle cell condition. Now, the last thing I wanted to talk about is what is the situation if you have parents that do not show any symptoms of a disease, but then all of a sudden the child will have some sort of syndrome or medical condition that's associated with the gene. How can I figure out if the parents were carriers and therefore pass the recessive copy to the child causing the disease, or it was the result of actual new mutation in the child that caused the medical condition. So again, the possibility is where the parents, let's say, the carriers, therefore the child became the person with the disease, or the parents were not carriers, but the child is still developed the condition because of a mutation in the sperm or the oocyte during the process of gamete formation and reproduction. So exome and what you can do is basically sequence the exome and the genome and compare the parental genetic information with the child. If you do see the pattern in one of the alleles of the parents, that implies that the parents were a carrier of the condition, versus if the sequence is only present in the child but not the parents, it implies that it was a de novo or a new mutation that is represented itself in the child. So the algorithm basically, if comparing the DNA sequence of the parents with the child to see if there is similarities between the genetic information. If it is, that basically implies that there was a carrier parents. But if the mutation is absent in the child, sorry, absent in the parents but present in the child, that means that it was a new mutation. So right here, let's take a look at this. The male pedigree in this case is show no symptoms, but Millie has the symptoms, which is the offspring. And then Millie can pass their autosomal to their next generations, where, sorry, not, I should say next generation. The way that Millie could have got that mutation is the parents being the carriers, which caused her to have that, or neither of the parents are showing any symptoms, and then that mutation was brand new in Millie, causing the Rett syndrome. And the only way that you can again figure out which of these two scenarios you're talking about is by sequences the genetic information and see if the mutation was present in the parents, then it would be the first scenario. And if the mutation is absent in the parents, then this was a brand new mutation that affected the child. I apologize. I know this was a very long PowerPoint, so thank you for bearing with me. It is a very important concept. It is a big part of your lecture exam, so make sure you guys understand it and visit me during office hours if you do have any questions about the concept. Thank you.